Hi, my name is Noha Agahimi. I'm a chemical engineering major at the University of Nebraska, and I will be presenting on genetic engineering. First, we will learn about DNA, then how it plays into gene expression, and finally, we will look at some applications of genetic engineering in the real world. DNA is fundamental to genetic engineering. DNA stands for deoxyribose nucleic acid, which is a fancy way of saying a combination of a phosphate base, a sugar, and a nucleotide. There are 4 nucleotides, denoted by the letters A, T, C, and G. These letters stand for adenine, thymine, cytosine, guanine. Adenine always binds to thymine, and cytosine binds to guanine. The order in which these base pairs exist is what makes strands of DNA unique. It is also what genetic engineers work to change as they modify DNA. To get an idea of how impressive genetic engineering is, first we have to understand how small a single strand of DNA really is. The diameter of a DNA strand is 2 nanometers, which is 1,000,000 times smaller than a single grain of sand. Despite how small DNA is, scientists are always working diligently to make cool new genetic designs. So now that we've talked a little bit about what DNA is, we can look at how DNA plays into the process of gene expression to control cell and eventually bodily functions. To help understand it, I want you to think about the process of writing a letter. DNA is like the person writing a letter. Once the letter is written, DNA hands it off to what is known as messenger RNA. mRNA acts as the mailman, delivering the message to the final recipient, the cell. This is a more technical schematic of genetic expression. DNA tells the cell to make a specific strand of RNA called mRNA. Then, the cell translates the information sent by the mRNA to create an amino acid chain, which then folds into specific arrangements to create proteins which act as the primary drivers of bodily function. Now that we've discussed both DNA and gene expression, we can take a look at the various ways that genetic engineering affects our everyday lives. Throughout history, selective breeding has been a primary way of creating new dog breeds, taking the ferocious wolf and making it into the docile house pet we're familiar with. This is a way of genetic engineering that manipulates natural mating patterns to create a desired organism. The same can be done for food, which we'll discuss more later. We can also create new DNA in a lab by splicing 2 strands together to produce a new protein. Here's a video that shows how. So here we're basically seeing a piece of DNA that was engineered in the lab being inserted into a bacterial strand of DNA, which is that unique circular shape called a plasmid. So as we can see, the pieces of DNA in this circular strand are cut to create 2 broken ends, known as sticky ends, where the new strand of DNA can be inserted. This is the DNA that was created in the lab. And then what's known as DNA ligase, an enzyme, is able to join the new DNA and the old DNA together to create a formed plasmid with a new strand of DNA. This is what's known as recombinant DNA plasmid. So now that we know a little bit about how genetic engineering looks in the natural world and then also in the lab, we can look at the different ways that genetic engineering impacts our lives. So modifying DNA strands is essentially what allows us to create cool and new products, like blue or yellow strawberries. What cool things do you think could be created using gene editing? Leave it in the comment section below if you come up with different ideas of what you think would be cool in terms of genetic engineering. Gene editing has also been used to modify produce and make it more plentiful. Apples and corn have all gotten significantly larger, as you can see in the pictures above. So we went from those really small apples and small cobs of corn to the really big ones that you guys are familiar with on Thanksgiving, or when you're making an apple pie and need a really big apple, or multiple really big apples. Another cool thing about genetic engineering is that it can help prevent species extinction. The banana, for example, is designed in a certain way genetically that a specific virus, because all bananas in the world are identical to one another, a specific virus, one virus, can destroy the entire population of bananas and make them go extinct. Hopefully, in the future, scientists will be able to manipulate the genetic makeup of bananas in order to make them immune to different viruses that could attack them and lead to extinction, which is great because we all love banana bread, and I personally would be really upset if bananas went extinct. There are many aspects of genetic engineering that are very different to one another, and because of this, there are many different kinds of engineers that can contribute to genetic engineering. The four listed on the slide are only a few of the many different kinds of engineers that work on different specific parts of the genetic engineering process, which is still very new and changing, so more engineers might be responsible for different aspects in the future as well. For now, mechanical engineers and biomedical engineers are especially important for designing the different technologies that we're able to analyze DNA and gene expression with, for example, microscopes. Polymerase chain reaction is a process that was designed by a biomedical engineer in order to rapidly replicate DNA that was already existing so that it can be studied and used effectively and multiple different times, which is super important to genetic engineering. Biomedical engineers and biological systems engineers, in addition to chemical engineers, are responsible for a lot of the stuff that we talked about, the modification of DNA, the designing of new recombinant plasmids, things that work with the actual modification of the nucleotides in the DNA itself is what a lot of these biomedical and biological systems engineers and chemical engineers are especially responsible for working with. Okay, so that concludes our discussion of what DNA is, how it plays into gene expression, and finally what the aspects of genetic engineering are in the real world that we can tangibly see, feel, and even a lot of the times eat. If you have any questions about the genetic engineering process, about DNA, about gene expression, leave it in the comments below and I will try and answer them as best as I can. You can also email the Engineering Ambassadors organization and ask your questions there. Thank you so much for listening. Now we're going to get into the activity that you guys will be doing in order to extract your own DNA from wheat germ. Hello everyone. So today we're going to be extracting DNA from wheat germ. So you start off with about half a teaspoon of the wheat germ in your conical tube. I have already put that in here, but you're going to want to take a couple seconds and do that. Feel free to pause this video at any time if you want to follow me step by step. So we have half a teaspoon of the wheat germ in the conical tube, which has the blue cap. And then we're going to want to add 20 milliliters of hot boiling water. So before this video, or before you attempt this experiment, please boil water because you're going to need that in order to extract your wheat germ. And use the graduations on the side, which you can't really see in the camera here, but there are graduations that you'll be able to see on your conical tube that will tell you how many milliliters of each thing you have, which will be super helpful in adding your various ingredients. So first you start with your 20 milliliters of water, which I just have in a cup right here, and I'm going to pour this real quick using my graduations to measure out 20 milliliters. Here I've got 20 milliliters. All right, so now I'm going to add about 1 milliliter of dish soap and then shake gently. The water is going to loosen that outside layer, the cell membrane and the cell wall, in order to give us access to that DNA. And then this is going to further loosen that cell membrane in order to give us access to DNA. So I'm just going to use graduations to do about 1 milliliter. And then I'm going to shake this. I'm going to shake gently for about 2 minutes. Okay, so I have shaken this for approximately 2 minutes. As you can see, you're going to have that layer of white foam, and then you're going to have like an intermediate layer, and finally your wheat germ. And then what you're going to want to do is you're going to want to remove that top layer of white foam. So what I like to do is just take a pencil and kind of dig it out. You're not going to want to pour it out because then the actual liquid is going to come out with it, and we don't want that because that's what we're going to precipitate our DNA out of in just a second. So go ahead and remove that white foam. So once you have that white foam removed, you're going to add as much as you can. Remove as much of the white foam as you can. There's a much less white foam if you shake it gently. I didn't shake it so gently, so I had quite a bit. But remove as much as you can, and then you're going to want to add 10 milliliters of rubbing alcohol. This is your final step, and you're going to want to add this 10 milliliters very slowly. This is the most important step because this is what's going to precipitate our DNA out of the liquid that we formed here. Alright, so once you've added that 10 milliliters of rubbing alcohol, in between the top of your liquid and then that second layer, you're going to see white floaty things. That is your DNA. And congratulations, you've extracted DNA from wheat germ. If you'd like, you can take a pencil or a stick and lift those out of there if you want a closer look. But that is all. Thank you for joining me during this experiment and for the presentation. Have a lot of fun, and send pictures of the wheat germ extraction that you complete to the UNL Engineering Ambassadors email. Thank you very much.